Alright, so we're going to start by drawing out the sarcomere. So for our sarcomere, we're going to have a Z line. We're going to have two Z lines, and that's just the space between the whole sarcomere. Then we have our thin filaments, or actin filaments. We have, actually draw those in a different color. So here's our actin filaments. We'll have our thick filaments, which will be our myosin filaments. So myosin, actin, our Z line, which is the boundary of our sarcomere. And then there's the H zone, which is just this space where we don't have any thin filaments, but we have our thick filaments. And then we have I bands, which would be all of this space, any space that appears light in the sarcomere will be our I band. Then we have our A bands, which are darker regions. And that is our sarcomere. So actin will have all those active sites that we need when we get to the power stroke. And then myosin will have the myosin heads required for the power stroke. So in order for an action potential to cause a muscle contraction, we're going to go back to the synapse of the neuron onto the muscle cell. So here's our muscle cell. Actually, draw that a little bit smaller. So when we have an action potential up on our nerve, I guess this will be our motor unit. That's going to be the one nerve that innervates all of our muscle fibers that we want to work with. So we have an action potential travel down this nerve. It's going to cause all these neurotransmitters, these vesicles, to reach the synapse where the snare proteins can pull them in and release them in their neurotransmitters across the synaptic cleft. So those neurotransmitters are actually going to be taken up by their own channels, and that's going to continue our action potential along the membrane of the muscle cell. As that action potential travels down our T-tubule, that's actually going to stimulate our it'll stimulate the ryanodine receptors, which will actually uncork the sarcoplasmic reticulum and allow all this calcium that we have stored inside to be released through that ryanodine receptor, the DHP receptor that is also there. When that calcium is released, it's going to flow out and it's going to go to our sarcomere. So when it reaches our sarcomere, it is going to bind to troponin C. So that calcium is going to go down to the actin filament, and it's going to bind to troponin C over on the actin filament. That's actually going to move the troponin that runs along the actin filament away from the active sites. So now that we have exposed active sites, we can move towards our power stroke. So our power stroke, we're gonna have a myosin head on our myosin filament. And that myosin head is gonna have an ADP along with a free phosphorus. So at this point, it's kind of like ATP. That is gonna bind to our actin active site, and it's going to form a cross bridge. Now once it's formed the cross bridge, ADP and this phosphorus are going to come off of the myosin head. The energy released from this reaction is going to cause the myosin head to cock that way, moving the actin filament inwards, which is going to result in a contraction. Now, in order to reset the myosin head, we're going to put an ATP on here. And that ATP is going to allow us to drop the myosin head so that we can enter a relaxed state. And then we're going to discuss the various kinds of energy that are used for muscle contraction. So we have a few different options for energy when we want to get a muscle contraction. We have cytosolic ATP, the ATP that's already there in our muscle. Now that energy, it's really good for rapid, strong movements. We have about enough to get 1 to 3 seconds of energy out of it. Now, once we've depleted that cytosolic ATP, we're actually going to shift over to another molecule called creatine phosphate. Now, creatine phosphate is a really nifty molecule that's able to go to that depleted ATP, the cytosolic ATP that we had, and it's going to drop off this inorganic phosphate, and that inorganic phosphate is going to bind to the ADP and create ATP. So because we're able to do this, and we have this creatine phosphate that's really effective at replenishing our ATP stores, this is actually going to allow us to use that energy for about 1 to 10 seconds. That's going to allow us to sustain some hard movements that require a lot of energy. once we've depleted all of this, or we're requiring a much longer amount of energy store, we're gonna move over to glycolysis. So we're gonna consume glucose in order to keep on supplying energy to our muscles, and this can be done for about a minute. The problem with this is that it usually leaves us with lactic acid when we have to turn over enough energy. That lactic acid is going to build up, and over time, the lactic acid doesn't contribute to fatigue, but muscular damage will, and we're going to end up running out of energy stores. Lastly, we're going to move over to oxidative energy stores. So we're going to use oxygen in order to turn over ADP to ATP. And we can do this for a really long period of time. However, this leads to the most fatigue. And because it's at a slower rate, it's really only going to be used for events that do not require rapid use of energy. And that's it.